Okay, share screen. All right, so part 3. Okay, so we talked through all the steps of glycolysis and citric acid cycle and electron transport chain. Okay, in the last section. So this will be the third and final part. Okay. Okay. So just one key note point. Okay. So there's 2 conditions that you're struggling with. One is aerobic, when you have oxygen. So when you normally breathe, you have plenty of oxygen. But imagine the situation where you're like lifting weights or sprinting and stuff like that, or running for your life. Okay, all these conditions, your body isn't going to get enough oxygen. Okay, so if you don't have enough oxygen, your muscles go toward anaerobic respiration. Okay, without oxygen. Okay, so anaerobic respiration, you can go through glycolysis. Okay, easily, because none of the steps of glycolysis require oxygen. All that requires oxygen is in the electron transport chain, okay, and therefore in the mitochondria. Okay, so everything in the cytoplasm occurs without oxygen. Okay, so you can still do this. Okay, so I can, okay, get pyruvate. Okay, I get pyruvate, and I can use pyruvate, but what I need to do, when I don't have oxygen, is to recycle the NADH back into NAD+. So I take the pyruvate, okay, and because, where is my screen? Okay. Because I cannot do this part, this is the only thing I can do. Okay, I take the NADH and I move it back to NAD+, and so I reduce pyruvate to lactate. That's why you build up lactic acid in your bloodstream when you exercise intensely. Okay, and then I just reuse NAD+ for glycolysis again. Okay, so I get much less ATP than before. I only get 2 instead of the 30-something ATP. Okay, but it's possible to use. Okay, I don't need oxygen because all this stuff requires oxygen. Okay, all this stuff requires oxygen. Okay, all right. Okay. Okay, proteins are a key role to cellular function. Okay, so proteins do basically everything in a cell. Okay, there's a protein alphabet that's made out of 20 different amino acids. The sequence and the number of amino acids actually give rise to different proteins. So you can think of proteins as like words, but they're just words, but they're really, really long words. Okay, but the order of the letters, okay, and the number of the letters tells you what the word is and therefore what the protein does. Okay, so in our DNA, we have a whole bunch of nucleotide sequences, and we have to translate that into proteins. Okay, and so every 3 letters of RNA, I'm going to explain how we get this RNA in a little bit. Okay, but every 3 letters of this RNA, okay, every 3 letters of this RNA forms a single amino acid. You string together all these amino acids and you get one big long protein. Okay, so some definitions, genes, okay, the differences between a constitutively active gene, which is always made, and or a regulated gene, which is either turned on or off, depending on your needs. Okay. Okay. So transcription is to go from DNA to RNA. Okay. So when you transcribe something, so my analogy is this. Okay, you go to an old library. The old library back in the day does not allow you to check out books. Okay. What it does allow you to do is copy them. Okay. So you go in, you find your book, you find a piece of paper, and you copy that page. You don't want the whole book, you usually just want a page. Okay. You take that page and you copy that page. Okay. You take that page, you copy it. That's transcription. You're transcribing the page. Okay. After you come out of the library, you can translate that page into a language. Like if the page was originally in ancient Greek, you want to translate it in English, you can translate the thing. So you go from the language of nucleotides, the mRNA, into the language of proteins, okay, amino acids. Okay, and the last thing you do is the post-translational modifications. That's where all the phosphatases and aminotransferases come into play, and methylases, strengthening off, etc., etc. All those are post-translational modifications. Okay. All right. So here, okay, you have every step of the process. Okay. You turn on a gene. You make a copy of the mRNA. You modify the mRNA for processing. Okay. And then you pump that out of the nucleus, okay, through the nuclear pores here, and then you translate it into amino acids. Okay, now those amino acids will form a protein chain, which will form a protein, and then you'll have post-transcriptional modifications. Okay. All right, so DNA, the whole purpose of DNA is to guide the synthesis of RNA. Okay, so you need a template strand, you need an RNA polymerase, you need a promoter, you need transcription factors. Okay, so let me explain how this works. Okay, so the first thing you have is a RNA polymerase. Okay, so we have a DNA here. The DNA unravels where the RNA polymerase binds. Okay, so it unravels. Now the RNA polymerase, okay, RNA polymerase is a type of synthase that makes RNA. Okay, polymer, it makes more RNA. Okay, so I take the template strand, I copy it. Okay, I bind to the template strand and I make a copy of the template strand. Okay, there's a template strand. Okay, so for example, okay, white screen for a second. Okay. If the DNA is ATGC, CCAAG, C, C. Okay, so 1, 2, 3, 4, 5. Okay. Okay, so this is my template strand of DNA. So make sure you know how to do this. The next thing I need to do is translate this template strand into mRNA. Okay. So the way this works, okay, in mRNA, so A's bind to T's or U's. In the mRNA, there's no T's, there's only U's. So A's bind to U's, T's bind to A's, G's bind to C's, and so GGG. This is UU. And then GTA, so CA, G, and GGG, U, A. Okay, hopefully that makes sense to you guys. Okay, if not, we can practice it in lecture. In fact, I plan to do this in lecture a couple of times to make sure you guys really understand this. Okay. Okay. So now I have the mRNA, and then I have the splicing. Okay. The splicing is pretty simple. Okay. Let's draw it out. Okay. The splicing, I have regions of the DNA that will come out of the nucleus and regions that will stay in the nucleus. The regions that stay in the nucleus are going to be introns. The exons are going to come out of the nucleus. So the exons are usually drawn in blocks. The introns are usually drawn in lines. Okay. Okay, so I have 4 exons. Okay, I'm going to end up splicing them. So I'll splice them this way. And I can get 1, 2, 3, 4. Okay, and that will be my DNA sequence that I export. Sorry, my mRNA sequence that I export. Okay, if I can also make, okay, 1, 3, 4. Okay, 1, 3, 4. So these are the mRNA processing. I can also make 2, 4. Oh, 2, 4. Sorry. So 2, 4. So I can make multiple combinations of these guys, and so I can come up with one gene. I can actually make different proteins. Okay, these proteins will all be roughly the same, but they'll have slightly different things, and therefore they perform slightly different functions. Okay. All right, so that's mRNA processing. Oops. I did not want to do that. You want to do all this stuff. There we go. Okay. So the next thing is this part comes out. Okay. So this part here comes out of the nucleus, goes to the cytoplasm, binds to a ribosome, and now the ribosome is going to look at this in 3-letter chunks. Okay. C, G, 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 U, C, A, U, G, U, G, G, U, A. Okay. Okay. So the very first thing the ribosome looks for actually is something called a start codon. Okay. So now I'm going to jump back into the presentation for a second. Okay. And show you this is the codon map, okay, or the codon chart. Okay, so the first thing I'm gonna look for is the start codon. If you look all through this, you'll find the start codon right there, AUG. Okay, start codon. The first start codon will always be methionine. Okay, so I will always put in the methionine first. So let's go back to my whiteboard. I ignore everything that's not the first AUG. So all of this is not useful. Okay, so I look for the first methionine. So that is the first one. Okay, and now I break them up into three-letter blocks. Okay, each block is called a codon. Okay, the first one is methionine. Okay, the second one I do is look at this. So I'm looking for GGG. G. So first letter is G, second letter is G, third letter is G. So glycine. Okay, so I go back to my chart, put in glycine. Okay, now I find my next codon, UAA. So now I'm going back to my codon chart. UAA is a stop codon. So all I do then is stop. Okay, that's it. That is my amino acid. Okay, I look for the first start codon. I start methionine. I go until I hit the first stop codon. After that, I stop. Okay, it does not matter if there is amino acids nucleotides after the stop codon. Okay, none of these things matter. As long as I hit the stop codon, I stop. Okay, hopefully that makes sense to you guys. Okay, all right. Okay. So no, this is translation. Okay, I find the first AUG, and then I add amino acids until I hit a stop codon. Okay, so the last 2 minutes, I'm gonna put on camera off. We'll let you guys watch the video. Okay, unfortunately, I can't upload this video to Canvas because it's too big.
This student is cramming for her biology test.
Okay.
With only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine. This student is cramming for leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, hopefully that makes sense to you guys. All right. So after the making of the protein is a big long string. It needs to fold and then form links, and then some of them are cleaved or cut into like some parts are cut out, so it makes it work better. And then you add other stuff, and then you assemble them together to form polymeric proteins with different proteins coming together to form a bigger protein, so they can function together all at once. Okay, all right. So hopefully that all makes sense to you guys. Again, I'm always available during class to explain some of this stuff. If you guys don't understand, I think it's more useful if you guys watch it once, and then we can talk about things during class. That makes it easier. Okay. All right. Bye, everybody.